Welcome to the deep dive. Today we're tackling forensic DNA. It's that fascinating spot where genetics meets the law.
Exactly. And our mission here is really to chart the journey, how we went from some, frankly, quite shaky forensic methods,
Yeah, unreliable stuff.
right, to the gold standard we have today.
Yeah.
Something that can pinpoint someone at a scene, or, just as importantly, clear someone who's innocent.
Okay, so before about, what, 1986, things were pretty limited.
Very limited. I mean, you had your standard fingerprints, blood typing. Those were okay,
Okay, as far as they went.
as far as they went. But then you also had things like bite mark analysis, hair microscopy, highly subjective, often unreliable. No real way to say this biological sample is this person.
Right, so that sets the stage perfectly. A huge leap from that uncertainty to genetic near certainty. Where did that begin?
It really kicked off in 1985. A researcher named Dr. Alec Jeffreys, he discovered something called minisatellites.
Minisatellites.
Yeah, or as he termed them, variable number of tandem repeats, VNTRs for short.
Okay, VNTRs. But hang on, aren't these repeats in the so-called junk DNA? The parts that don't code for proteins? How did that become the key?
That's the brilliant part. It wasn't what the sequence was, it was how many times it repeated. The length varied massively between people.
So the number of repeats changes.
Exactly. These sequences, maybe 15 to 100 base pairs, they get repeated over and over, and one person might have, say, 10 repeats, while another has 30 or 50. The overall length of that chunk of DNA becomes very different. We're talking maybe 1 kilobase up to 20 kilobases long.
So you're measuring size differences, not the actual sequence.
Precisely. And the variability is just huge, even looking at just 4 different VNTR spots or loci.
Loci, okay.
And if each of those loci had maybe, let's say, 20 different possible lengths, 20 alleles,
Yeah.
you multiply that out and you're looking at over 2 billion possible combinations. Billions. Suddenly, you have a way to uniquely identify someone biologically.
That is staggering. Okay, so that was the concept. But the method back then, the VNTR method,
Yeah.
wasn't exactly quick and easy, was it?
No, definitely not. It was quite cumbersome. First off, you needed a fair bit of DNA. About 50 micrograms, something like 10,000 cells.
Wow, that's a lot compared to today.
It is, and the DNA had to be pretty much intact, not degraded.
So how did they actually do it? Can you walk us through that?
Sure. You'd extract the DNA, then cut it up using a specific restriction enzyme that snips the DNA on either side of the VNTR region.
Right.
Then you'd run those fragments on an agarose gel electrophoresis. That separates them based purely on size. Smaller pieces move faster.
Okay.
Then came the tricky part, southern blotting. Transferring that DNA pattern to a membrane and using a radioactive probe that would stick only to the VNTR sequences, you'd expose it to film.
And eventually get a pattern of bands, like a barcode.
Exactly like a barcode. Unique to an individual, mostly. But yeah, slow, needed lots of good quality DNA.
And the first big test case, the one that really showed its power, was the pitchfork case.
That's the one. 1986, Leicestershire, England. 2 separate rape murders. Police had a suspect, Richard Buckland, who even confessed to one.
Right.
But Jeffrey's VNTR profiling was used. It showed the semen samples from both victims came from the same man.
Okay, linking the crimes.
But critically, that profile did not match Richard Buckland.
Wow. So it exonerated him.
Immediately, first-time DNA profiling led to an exoneration. Eventually, they used it in a massive voluntary screening, found the real killer, Colin Pitchfork, and convicted him. It proved the technology worked both ways.
Catching the guilty, freeing the innocent. Incredible. But as you said, needing 10,000 cells.
Yeah.
That's a problem for most crime scenes, right? Trace evidence, a tiny blood spot, a single hair.
Useless for VNTRs, generally. You needed a visible stain, really. That was the major limitation.
So what changed? What allowed forensics to move beyond needing large samples?
The invention of PCR polymerase chain reaction, that was the absolute game changer.
The DNA photocopier.
Essentially,
Right.
yes. PCR lets you take a tiny, tiny amount of DNA, maybe from just the root bulb of a single hair, or even from really old degraded samples like ancient bone. And you can amplify just the specific bits you're interested in, make millions, even billions of copies in just a few hours. Suddenly, sample size wasn't the barrier it used to be.
Okay, so PCR solves the quantity problem. And did that lead to using different DNA markers too? Away from the long VNTRs?
It did. We shifted to short tandem repeats, STRs, sometimes called microsatellites.
STRs, shorter repeats.
Much shorter. The repeated unit is only 2 to 9 base pairs long, and they're repeated maybe 7 to 40 times. So the whole STR locus is much smaller than a VNTR.
Example.
Take the D8S11179 locus. The repeat is just 4 bases, TCTA, and a person might have anywhere from 7 to 20 copies of that TCTA repeat. Much smaller target, much easier to amplify reliably with PCR, especially from degraded DNA.
Makes sense. And I imagine for this to work across different labs, different countries, everyone needs to be looking at the same STRs.
Oh, absolutely. Standardization is crucial. In the U.S., the FBI established a core set of loci. Originally 13, now it's 20 STR loci that every forensic lab analyzes.
20 specific locations in the genome.
Right. Most European countries use a standard set of 12, but there's a lot of overlap. And the analysis is streamlined now with commercial kits.
Kits, like prepackaged tests.
Pretty much. These kits have everything needed. Primers designed to flank each of those core STR loci. And importantly, the primers for different loci are tagged with different colored fluorescent dyes.
So you can tell them apart. Blue for one locus, green for another.
Exactly. Blue, green, yellow, red. This lets you amplify all 20 loci together in a single PCR reaction. It's called multiplexing.
Efficient. So after PCR copies all these STR fragments, how do you measure their lengths? Still using gels?
Not flat gels anymore. We use capillary electrophoresis, or CE.
Capillary, like tiny tubes?
Very thin glass tubes, yes, filled with a gel-like polymer. The amplified DNA fragments, tagged with those fluorescent dyes, are injected into the capillary, and electric current pulls them through.
And shorter ones move faster, just like in the old gels.
Same principle, just much faster, more automated. As the fragments pass a detection point near the end of the capillary, a laser excites the fluorescent dye.
And a detector reads the color.
Reads the color and measures the exact time it took to arrive, which translates directly to its size, meaning the number of repeats. Software plots this data.
What does that plot look like?
It's called an electrophoregram. You see a series of peaks against a baseline. Each peak represents a DNA fragment of a specific size and color.
And how does that tell you the genotype?
Well, remember, you inherit 1 chromosome from your mom, 1 from your dad. So for each STR locus, you have 2 alleles, 2 copies.
Right.
If those 2 copies have different numbers of repeats, say you got 15 repeats from mom and 18 from dad, you're heterozygous, you'll see 2 separate peaks for that locus on the electrophoregram.
Okay, 2 peaks for heterozygous. What if they're the same?
If you inherited the same number of repeats from both parents, say 15 and 15, you're homozygous, then you just see 1 peak, but it's usually about twice as tall because both alleles are contributing to it.
Got it. And the profile itself is just recorded as the number of repeats, like D5S13581518.
Exactly that. A string of numbers representing the alleles at each of the 20 core loci. That's the DNA profile.
Okay, so that's the standard STR profile. But the source material also mentions specialized techniques for trickier samples. What's first?
Y-chromosome STR profiling, using STR markers found only on the Y chromosome.
Why would you need that?
Think about a sexual assault case. You might have a vaginal swab with a mixture of female victim cells and male perpetrator cells. Lots of female DNA, maybe very little male DNA.
Right, the standard STR test might just pick up the victim's profile.
Correct. But Y-STR analysis specifically targets the Y chromosome, so it only amplifies the male DNA present. It helps isolate the contributor profile in mixed samples.
Useful. Any drawbacks?
Big one. The Y chromosome is passed down largely unchanged from father to son through the male lineage. There's very little recombination, meaning fathers, sons, brothers, paternal uncles, paternal grandfathers. They all share the same Y-STR profile. It can tell you DNA came from a male in that family line, but it can't distinguish which male.
I see. Good for inclusion-exclusion of a family line, but not individual identification within it. What else?
Then there's mitochondrial DNA, mtDNA, profiling.
Mitochondria.
Yeah.
The powerhouses of the cell. They have their own DNA.
They do. A small circular chromosome. And the key advantage here is copy number. Each cell has only 1 nucleus with 2 copies of each regular chromosome, but it can have hundreds, even thousands of mitochondria.
So thousands of copies of mtDNA per cell.
Exactly, which makes mtDNA analysis fantastic for really, really challenging samples. We're talking tiny amounts, very old samples, or highly degraded ones like skeletal remains from mass disasters like 9/11 victims or ancient bones.
Because there's just so much more starting material.
Right. But it also has an inheritance limitation. You inherit your mitochondria and thus your mtDNA exclusively from your mother.
Only from the mother.
Only from the egg cell. Sperm mitochondria don't usually enter the egg. So you share the exact same mtDNA sequence as your mother, your siblings, male and female, your mother's siblings, your maternal grandmother, and so on up the maternal line.
So like Y-STRs for the paternal line mtDNA can't distinguish between maternal relatives.
Correct. High sensitivity, but lower discrimination power than nuclear STRs.
Okay, one more specialized type mentioned.
SNP profiling.
Single-Nucleotide Polymorphism.
SNPs, yeah. These aren't repeats. They're just single base pair changes at specific points in the genome.
Yeah.
Like at a certain position, most people might have a G, but some have an A.
These are common.
Very common. Occur maybe every 500 to 1,000 nucleotides throughout the genome.
Why use SNPs if STRs are the standard?
Degradation. Extreme degradation. Because the SNP is just a single base change, the piece of DNA you need to amplify to detect it can be incredibly short, maybe only 50 base pairs long.
Ah, so even if the DNA is chopped into tiny fragments where STRs are too broken to analyze,
you might still be able to get SNP information. The downside is that most SNPs only have two common versions, two alleles, like the G or A example. Unlike STRs, which can have 20 or more alleles.
less variable.
Individually, yes.
Yeah.
So you need to analyze a lot more SNPs, maybe 50 or more, to get the same statistical power, the same ability to discriminate between individuals as you get from the 20 core STR.
Okay, that makes sense. Now, these SNPs, they're also the basis for something quite new and controversial, DNA phenotyping.
That's right. This is a big shift. Phenotyping isn't about matching a suspect's DNA to crime scene evidence directly.
What is it about then?
It's about predicting what the source of the DNA might look like, using patterns of specific SNPs that are known to be associated with physical traits.
Like what kind of traits?
Things like eye color, hair color, skin pigmentation, even biogeographical ancestry, where in the world someone's ancestors likely came from.
Seriously, how accurate is that?
It's getting surprisingly accurate for some traits. The chapter mentions, for example, that analyzing just 6 specific SNPs can predict whether someone has blue or brown eyes with about 95% accuracy.
Wow.
And maybe 22 SNPs can predict black hair color with 90% accuracy. Skin color and ancestry prediction are also areas of active research.
The book gives the Bouzigard case as an example of this in action.
Yeah, that's a striking case. Police were investigating a murder, and initial leads pointed towards a suspect of Hispanic descent.
Okay.
But DNA phenotyping was done on the crime scene sample. The prediction came back: likely pale skin, blue or green eyes, brown hair, and crucially, Northern European ancestry.
Completely different from their initial theory.
Totally different. They basically had to reset their suspect profile based on the DNA prediction. They generated a new composite sketch based on the phenotype predictions.
And it led to an arrest.
It did. Led them to a suspect who matched the phenotype, and his STR profile eventually matched the crime scene evidence. It generated a lead where traditional investigation had stalled.
That's undeniably powerful as an investigative tool. But you can immediately see the ethical flags, can't you?
Oh, absolutely. Huge ethical concerns.
Using genetics to predict appearance, ancestry, it sounds like it could easily slide into or reinforce racial profiling by law enforcement.
That's the #1 concern. Plus, privacy issues. And it's important to stress this technology is not currently validated or widely accepted for use in court proceedings.
So it's an investigative lead generator, not evidence for conviction.
Primarily, yes. And it's treated with extreme caution because of those potential societal harms and the risk of error or misinterpretation. It shifts forensics from identifying a known suspect to potentially creating a suspect profile from scratch based on genetics.
Right. Okay, let's shift back to standard STR profiles. When you do get a match between a suspect and crime scene evidence, that's not automatically proof, is it? It's about probability.
Exactly. A match doesn't mean this is the person. It means this person cannot be excluded as the source. The crucial next step is calculating the significance of that match.
The random match probability.
Precisely. What is the probability that a random, unrelated person picked by chance from the population would happen to have the exact same DNA profile?
How do you calculate that?
Using population databases. Forensic labs maintain databases showing how common each specific STR allele, like 15 repeats or 18 repeats at a locus, is within different population groups.
Okay.
Then you use the product rule. You find the frequency of the genotype observed at the first locus, say the frequency of having 1518 at D5S3358. Individual locus genotype frequencies together, because the inheritance of alleles at different STR loci are generally independent events.
It's like flipping multiple coins.
Kind of, yeah. The chapter gives an example using just 5 loci. Multiplying those frequencies might give you a combined profile probability of something like 9 times 10 to the -7.
Which is?
About 9 in 10 million. So, the chance that a random unrelated person would match that 5-locus profile is incredibly small.
And that's just 5 loci. The U.S. standard uses 20. What happens to the probability then?
It becomes astronomically small. The source quotes a probability for the 20 core loci profile as being on the order of 1 times 10 to the minus 28.
1 followed by 28 zeros after the decimal point.
Yeah, I mean, it's a number so small it's hard to comprehend.
It basically means the profile is expected to be unique across the entire human population, past and present. For practical purposes, it's considered individualizing.
But there's a catch, right? You mentioned unrelated people.
Huge caveat. The standard product rule calculation assumes the suspect and the random person are unrelated. But close relatives share significant amounts of DNA.
Identical twins.
Identical twins will have the exact same STR profile. You cannot distinguish them with this method.
What about siblings? Or parent-child? Siblings share, on average, 50% of their alleles. Parent and child also share exactly 50%. So the probability that a sibling or parent would match is much, much higher than for a random person. Statisticians have to use different calculations that account for that shared ancestry if relatedness is a possibility.
Okay, so these powerful profiles, once generated, they need to be stored and compared. That's where databases like CODIS come in.
Right. CODIS stands for the Combined DNA Index System. It's maintained by the FBI.
What's in it?
It has several parts, but the main ones are the convicted offender database profiles from people convicted of certain crimes.
Okay.
And the forensic index profiles developed from crime scene evidence where the perpetrator is unknown.
So you can compare new crime scene profiles against convicted offenders or link different crime scenes together.
Exactly. A new crime scene profile goes in, gets searched against the offender database for a potential suspect, a hit, and also searched against other forensic profiles to see if this perpetrator has struck before.
How big is CODIS? How effective?
The numbers are pretty staggering. As of 2016, the book states CODIS contained over 16 million profiles.
16 million. Wow.
Yeah, and it had aided in over 350,000 investigations by that point. It's undeniably a powerful tool for law enforcement. But of course, its very existence raises privacy concerns.
We'll get to those. But first, even with these amazing tools, things can go wrong technically, right?
Yeah.
It's not foolproof.
No, definitely not. There are technical issues. Labs can have backlogs. Samples can be degraded or contain inhibitors that mess up PCR. And then there's human error.
Like mixing up samples.
Absolutely. Sample switching, contamination in the lab. These things can happen, though labs have strict protocols to minimize them.
Yeah.
But one really concerning issue highlighted is DNA transference.
Transference. You mean DNA moving from one place to another?
Yes, indirectly. The Lukis Anderson case described in the text is just chilling.
What happened there?
Anderson was a homeless man picked up by paramedics and taken to the hospital for intoxication. He had a solid alibi. He was physically in the hospital, monitored when a millionaire was murdered elsewhere in town.
Okay, so how did his DNA get involved?
His DNA was found under the victim's fingernails. A perfect match to Anderson from the CODIS database. He had a prior conviction. He was arrested and charged with murder.
But he was in the hospital. How?
It turned out the same paramedics who treated Anderson later responded to the murder scene. They must have inadvertently picked up Anderson's DNA, maybe just skin cells on their equipment or uniforms. He was known to shed a lot of DNA and then transferred it to the victim's body during their response there.
Wow. So the DNA was real, the match was real, but the interpretation that it meant he was there was completely wrong.
Exactly. It highlights just how sensitive DNA detection is, how easily DNA can be transferred, and the critical importance of context in interpreting a match. Anderson was eventually cleared, but only after months in jail.
That sensitivity also opens up another scary possibility: deliberate tampering.
Yes. The fact that STR profiling is so sensitive and based on amplifying specific short sequences means it's theoretically possible to create fake DNA evidence.
Oh.
Researchers actually did this. They synthesized short DNA fragments corresponding to the alleles of a known STR profile. They mixed these synthetic fragments into someone else's saliva or blood, and standard STR analysis generated the target profile, effectively planting false evidence.
So you could potentially frame someone.
Theoretically, yes. The source suggests a potential countermeasure for the future might be looking for epigenetic markers, like DNA methylation patterns. Natural DNA has these patterns, but synthetic, manufactured DNA wouldn't.
A way to tell real from fake.
Yeah.
Okay, this all leads us squarely into the ethical minefield surrounding forensic DNA.
It's a complex area, definitely. The chapter flags several key ethical debates.
What was the first one?
Collection and storage. Who should have their DNA profiles stored in databases like CODIS? Just convicted felons, arrestees, everyone. And under what circumstances can police collect DNA? Do they need a warrant, or can they collect abandoned DNA from, like, a discarded coffee cup without consent?
Big questions about privacy and government surveillance there. What's next?
Familial DNA searching. This is where investigators search the database not for an exact match, but for a partial match.
Why would they do that?
A partial match might indicate that a close relative of the person who left the crime scene DNA is in the database. So police might investigate the family members, brothers, fathers, sons of the person who partially matched.
So you're essentially putting people under investigation because their relative committed a crime,
Right.
even though they've done nothing wrong themselves.
That's the ethical dilemma. Is it a valid investigative-led technique or an unacceptable intrusion into the privacy of entire families, effectively creating genetic informants without their consent? It's highly controversial.
And the third major ethical issue.
That brings us back to DNA phenotyping. Should law enforcement be using genetic predictions about appearance and ancestry, given the high potential for reinforcing bias, racial profiling, and errors? Where do we draw the line between a helpful lead and genetic stereotyping?
Yeah, these aren't easy questions. Balancing the power of the science against individual rights and potential misuse.
It's a constant balancing act.
So let's recap this journey. We started with pretty unreliable methods pre-1986. Then VNTRs gave us the first real DNA fingerprint, limited by sample size.
Then the PCR revolution enabled STR analysis, the current gold standard, offering incredible sensitivity from tiny samples. We have standardized kits, capillary electrophoresis.
Right, leading to those highly discriminating profiles, and specialized techniques like YSTRs and mtDNA for specific situations like mixed samples or ancient remains.
And SNPs for extremely degraded DNA, which also opens the door, controversially, to phenotyping.
And critically, the interpretation relies on probability, the random match probability calculated using the product rule, giving us those near-certainty statistics for unrelated individuals.
Plus, the CODIS database system for storing and comparing these profiles, aiding countless investigations but raising privacy concerns.
And we have to remember the potential pitfalls: technical errors, human mistakes, the risk of transference, deliberate tampering, and the major ethical debates over collection, familial searching, and phenotyping.
It's been a remarkable scientific journey in just a few decades.
Absolutely. So, looking ahead, what does this mean for the future, especially as science keeps advancing?
Well, one fascinating point the chapter raises, almost as a final thought, touches on the very foundation of DNA profiling. The assumption that each person has one unique, consistent genome.
What could challenge that?
The source mentions chimerism and mosaicism. Think about people who've had organ transplants or bone marrow transplants. They can carry cells with two distinct genomes of their original one and the donor's.
Right. Tissue from someone else.
Or even after pregnancy. A mother might retain some fetal cells for years containing the child's DNA. This state of having mixed genomes from different individuals is called chimerism. Somatic mosaicism is when different cell lines within one person develop slightly different genomes due to mutations after conception.
So people walking around with more than one DNA profile inside them.
Potentially, yes. And as medical procedures like transplants become more common and our understanding of mosaicism grows, you have to wonder,
how might the increasing prevalence of people living with two distinct genomes complicate our current methods? Methods that are built entirely on the idea of finding one unique profile at a crime scene.
Exactly. How do you interpret a mixture if it originates from within a single individual? It's a complex biological reality that could pose a fundamental challenge to forensic DNA interpretation down the road. Something for you to think about.